Welcome dear students. In this video I'll be going to talk about the second step of glycolysis, that is the conversion of glucose 6-phosphate to fructose 6-phosphate. Now this process is actually reversible. The first step of the glycolysis is irreversible. Now this step is reversible and the product is fructose 6-phosphate. Okay, so here the enzyme that is responsible for this conversion named glucose 6-phosphate isomerase. You can see the product name is glucose 6-phosphate and the enzyme name is glucose 6-phosphate isomerase. That means this enzyme can make the isomer of glucose 6-phosphate. So in this step we have to understand a very basic thing about the carbohydrate molecules, that is the isomer. You have to understand what is isomer. A glucose is a monosaccharide. It has two isomers like galactose and fructose. All these three molecules have the same formula, that is C6H12O6. All the three molecules contain 6 carbon, 12 hydrogen and 6 oxygen. Now they are called isomer. I will tell you that the glucose and the galactose isomer are actually stereoisomer, but the glucose and the fructose is actually structural isomer. So why is the difference between stereoisomer and structural isomer? So to understand this I have to draw the linear structure and the ring structure of all these three molecules. So let me draw. Let's suppose this is a carbon, so there are 6 carbon. So this is actually the linear structure of this molecule, glucose. So there are 6 carbon, like this is the first, this is the second, third, fourth, fifth. So there will be another carbon. Okay, this is the linear structure of glucose. Okay, now you can see here that the glucose has a aldehyde group, CHO, aldehyde group. So the glucose is actually called aldose. If I draw the linear structure of galactose, it will look like same because it has all the molecules, but somehow it differs from the glucose by stereoisomers, that is called mirror image. So let me draw the galactose, CHO bond. Okay, the difference will be here in this 4th carbon and the all same. Let me draw the number. So this is the first, second, third, fourth, fifth and sixth. Okay, good. So now this is the glucose, this is the galactose, and I will also draw the fructose. At first I want to show you why these glucose and galactose are called stereoisomers. If you see here, the 4th carbon of the glucose and the 4th carbon of the galactose, they contain same atoms, but the 3D organization of that atoms are different. They are the mirror image of each other. So these are called stereoisomers. So glucose and galactose are stereoisomers. All the molecules arrangement are same. Now this is also, it has also the aldehyde group. So a galactose is also a aldose. But here we will focus fructose, the structure of fructose. Yeah, I can draw the ring structure of these two molecules. So that will be like this. So here H, OH, H, OH, this is the ring structure. You can see here, I can write the number first, second, third, fourth, fifth and sixth. And this is the glucose. The structure of the galactose will be same, but the difference will be here. I have drawn here the fourth number of carbon. So in case of galactose, the orientation will be opposite. OH will be on the top and H will be on the lower side. So here is only the difference between glucose and this is the galactose. The structure will be the galactose. They are stereoisomer. Now if I draw the structure, linear structure of fructose, you will see the structure is like that. The structure will be like this. Here you can see a difference, not in the you can say the orientation of the molecule or atom, but here is the difference is it has a ketone body, ketone. So ketone group. So it is actually, we call it ketose. So fructose is actually a ketose. You can see here there is a ketone group. All the carbon, like this is the first, the second, third, fourth, fifth and sixth. You can see here all the orientation of the carbon is actually same here, but here is a difference in the aldehyde group and the ketone group. Now this is called ketose. If I draw the molecular structure or ring structure of this fructose, you can see that it looks like this. Okay, here CH2OH, OH, OH, H, H, OH. This is an alternative. You can see here that it contains first, second, third, fourth, fifth and sixth. The structure of this glucose and the fructose is so different. So this is actually structural isomer. It differs in the aldehyde and the ketose group. Now we have seen here that glucose 6-phosphate is converted into fructose 6-phosphate. So here the hydrogen will be removed and a PO3 - 2, a phosphate group will be there. So this is the glucose 6-phosphate. Now in case of fructose 6-phosphate, the structure will be like this, -2. So in this video I have shown you that how this glucose 6-phosphate isomer, the name implies isomer, it make the glucose 6-phosphate into fructose 6-phosphate. Another information I want to give you for this enzyme, that this enzyme can be secreted out of the cell. It acts as this glucose 6 isomerase can act as a neurotrophic factor. It helps in the survival of spinal motor neurons. Also it acts as another functions. These are all information for your preparations. Neurotrophic factors, and it also acts as a factor that is actually helps in the metastasis. This glucose 6-phosphate isomerase can also be secreted by cancer cells and helps in the metastasis. Okay, so this is the second step of glycolysis. I will take a separate class for the step 3. Thank you for watching and always keep learning.